Hi, my name is Soumya Pillai, and in today's episode of Pure Science, we are diving into one of the most mind bending breakthroughs in modern biology. A study that is rewriting what we thought was impossible in human reproduction. Scientists in the United States have managed to create early stage human embryos, not from sperms and eggs, but from skin cells. Yes, you heard that right, from human skin cells. It is a discovery that could not only reshape fertility treatment by helping people who cannot have children through the natural processes, but also spark some of the biggest ethical debates in science today. In simpler terms, imagine this as being able to create life not from sperm and egg, but from a patch of skin. Because that's what scientists have managed, turning ordinary human skin cell into early stage embryos in a lab. But while this has stirred tremendous interest, it is also one of the most controversial steps in modern biology. Not because anyone is trying to make a baby this way, but because it could change how we understand fertility, reproduction, and even the origins of life itself. Now, let's get into how this was done. The researchers started with skin cells from a set of adult volunteers. Each of these cells already carry a full set of DNA with 46 chromosomes, the same genetic blueprint that makes up an entire person. They also took donated human eggs from female volunteers and carefully removed the egg's own nuclei, which is part of the cell containing their DNA. This left them with what can be visualized as an empty eggshell. The next step was to insert the nucleus from the skin cell into the empty egg. This process is a bit like what scientists did decades ago to clone Dolly the sheep, where they replaced the genetic material in an egg with the DNA from a body cell. But in the case of humans, this is only the beginning of the challenge. A normal human egg cell has only half the usual amount of DNA with 23 chromosomes, so that when it joins with sperm, which carries the other 23, the resulting embryo has the full set of 46 chromosomes. But the skin cell nucleus they just inserted contained all 46 chromosomes. So the scientists had to find a way to make this new hybrid cell forget that it had once a skin cell and behave like an egg, which could divide its chromosomes in half. To do that, they developed a special process called the mitomeiosis, which is basically a kind of laboratory trick that combines the features of two natural processes, mitosis and meiosis. It forces the cell to mimic what happens naturally when an egg is formed in the ovary, that is, splitting the DNA so that one half remains. In theory, that gives you a true egg again, but made entirely from the DNA of a skin cell. Once the step was complete, they fertilized the reconstructed eggs with sperms, just like in standard IVF. And something astonishing happened. A few of these eggs began to divide. They started developing into early embryos, tiny balls of cells that looked just like naturally formed embryos about 5 or 6 days after fertilization. In a normal pregnancy, that's the stage when an embryo would be ready to implant in the uterus. In this experiment, scientists stopped there. These embryos were not implanted, and the experiment was ended after a few days. But the fact that it worked at all is extraordinary. It proves that, at least in principle, you can create an embryo using just skin cell DNA and sperm. If this is perfected, it has the potential to alter the course of fertility treatments. Women who can no longer produce viable eggs due to age, chemotherapy, or certain genetic conditions might be able to have children using their own skin cells. In theory, it could even make it possible for same-sex couples to have a baby genetically related to both partners by turning skin cells from one partner into eggs and fertilizing them with the other's sperm. But that's still a long way off. The vast majority of these lab-made embryos had made major chromosomal errors, missing or duplicated chromosomes, or ones that didn't line up correctly. Out of all the attempts, only a small fraction developed into early embryo stage. Now, in natural reproduction, the process that halves the DNA is tightly controlled, but in the lab, it's still messy and unpredictable. So, this experiment is proof of concept, not a practical method. And then, of course, there are the ethical questions, perhaps even bigger than the scientific ones. Should embryos created entirely in the lab be treated the same as natural ones? How long should scientists be allowed to grow them? And who decides what uses are acceptable? Would it just be used for helping in fertility, studying early development, or something more controversial? There are also fears about the misuse of such technology. Could it one day be used for genetic selection or for creating the so called designer babies? The researchers, however, are clear. According to them, at this stage of research, this work is not about creating babies. It is about understanding the earliest stages of human life, how cells organize themselves, how genetic errors arise, and why some pregnancies fail so early. By studying these embryos, scientists hope to uncover insights that could improve IVF success rates and also prevent inherited disorders. But it all depends on how the next phases of the research pans out. That was all from me. I am Soumya Pillai and you were watching Pure Science.